Hey everybody. Um, so I wanted to try to do an Edpuzzle today and record my lecture like this instead of just lecturing you in class. I want to see if you like it more. So just doing a little bit of experimentation with some tech stuff. Anyway, so today we're going to be talking about the cell membrane because this unit is about how cells can transport materials into and out of the cell. And the cell membrane has a has the is really the thing that does that. That is the cell membrane's job, right? So in order to understand how it does that job, I want you to really understand the structures within the cell membrane first. So, let me present. Okay, can I move this? Beautiful. All right. So before we even get started, let me remind you again what this picture is showing you. Hopefully, you guys already recognize this as a cell membrane. A cell membrane is made up of all these little, they look like orange balls, right? Attached to these tails on the inside. The orange balls are charged. They have some kind of charge to them, right? So they're going to mix well with water because they are polar, and so is water. Water is also polar. But the inside here, all of the tails on the inside, that's actually non-polar. It's kind of like an oily material. And for anyone who has ever mixed water and oil before, you know that they don't mix. So water cannot pass through the middle here, which is why, as we've already said in a prior lesson, water needs a special protein channel called an aquaporin, a special passageway to move through the membrane. Anyhow, let's move on. So that this cell membrane, this thing here, the balls on this side and the balls on this side attached by these tails, this is actually called a lipid bilayer, right? This word up here, lipid bilayer. Um, really, these orange balls that I keep referring to are actually phosphate groups, right? And the tails are just a chain of hydrocarbons. What you need to know though is that the phosphate groups are polar and the hydrocarbons on the inside are nonpolar, right? And that's why water can't pass through it. So lipids are a category of a molecule that include, as we've gone over this before, fats, oils, and waxes. Remember, lipids are one of our macromolecules. We've learned that already. Okay. And the cell membrane is made of a flexible double layer of phospholipids, which is uh, the word I used earlier, right? Cool. So there are also cell surface structures, proteins inserted into the lipid bilayer that have different functions. This purple thing here, this purple thing over here, this orange thing in the back, this green thing, these are all different types of proteins that have different jobs. This protein right here is a protein channel. It's going to help transport materials into and out of the cell. This protein here, it doesn't really say what it is, but I imagine it could possibly be another type of transport protein called a not a protein channel, but I'll think of it later. Anyway, but its job is also to transport materials into and out of the cell. And then this protein in the back looks like it is likely some kind of receptor protein to receive signals of some sort. Um, and we're going to talk about all of those in this lesson. So, phospholipids, which are the orange head with the two tails, have a phosphate group, the head, that can mix with water at one end and two long lipid tails that do not mix with water at the other end, like this. So there is the head. It can mix with water because it is polar. And there are the tails. They are more oily. Okay, they don't mix with water. Those are non-polar. Right? So when water being on the outside of the cell, these polar phosphate head groups are going to be attracted to that water, and so they're going to end up lining up and touch the water. The cytoplasm is also mostly water, though. So what ends up happening is these phospholipids make they go back to back and they make a double layer because the heads are cool with touching the water, and the tails are not. Right? So they end up making a safe space for those tails. You know. So the cell membrane regulates what enters and leaves the cell. And a lot of when scientists first discovered this, they referred to it as a fluid mosaic model, because when they were looking at the cell membrane, they saw that the proteins, all those proteins that are on it, the channels and the receptors, and the markers, they don't stay in one spot. They actually float around the cell membrane, and it reminded them of a mosaic. There's a link here. If you hover over, if you let me see. You know what? I'll post this link in a comment on the Edpuzzle. I recommend that you go ahead and watch this video. Okay, I'll make the link available to you on the Edpuzzle. All right, watch the video so you can see what I'm talking about. And this is just a picture of what a mosaic is. If you don't know what a mosaic is, this is a mosaic. Those that discovered the cell membrane and its functions thought that it resembled this. Back to the cell surface proteins. Back to those proteins along the cell membrane. So some of them are channels, like this one. Could be an aquaporin. It could be a different kind of protein channel to make a passageway for other type of materials. All right, these transport proteins are also known as carrier proteins. That's the word I was thinking of before that I couldn't think of. So they could be known as carrier proteins or channel proteins, depending on what they look like. This is a channel protein. It just goes straight through. These allow larger things to pass through the cell membrane. That hexagon that just passed through, that's too big to pass through these phospholipids, so it needs this bigger gap, this bigger channel to pass through. That's what the protein is for. And these other things are the carrier proteins. So these are also, they have the same job. They just move things into and out of the cell, but in a different way. They will be made for very specific materials, and will only activate when its specific material enters it. So we just talked about how larger materials are able to enter and leave the cell through these proteins, right? But we didn't talk about small stuff. What about things that are even smaller? How do you think those are going to cross through the cell membrane? Before moving on to the next slide, I want you to stop it here and kind of think about it. Pause the video for a moment and try and come up with the answer for yourself. If you thought of this, congratulations. If not, it's okay. And if you just skipped ahead and didn't think about it yourself, then shame on you. Anyway, so like smaller molecules like oxygen gas, carbon dioxide, nitrogen gas, steroids, fat soluble vitamins, urea is part of urine, P, glycerol, some alcohols and ammonia, or many examples, are small enough to just go straight through the phospholipid bilayer, just right through it. All right. That tells us that, and well, that's partially they are able to do that because these things are not nonpolar or they don't have so strong a charge that they get attached here. They don't get held up anywhere, right? Like water would. Water's not going to pass through. One, because it's polar and it's not going to mix with the oily inside, and two, because it's probably going to make some kind of hydrogen bond with the phosphate head here, right? So those smaller substances, they can just squeeze right through, as I just talked about. Just like that. See that little white thing moving right through? Nice. You might be wondering what the heck we're looking at right now. So if we were to get a really close-up look at the cell membrane, uh, these would be the top of the phosphate heads. And we would see small gaps in between them. And that's how these smaller molecules like oxygen gas and other things can fit through. They squeeze right through those little gaps. Right. So we talked about our transport proteins, right? Things that help bigger materials move into and out of the cell. The other kind of protein that we're going to find along the cell membrane is a receptor protein. These things will receive signals, right? They will intercept, right? Think of that. So those of you that are football fans, what's an interception, right? An interception is when a player intercepts the ball, right? Receives it. You can kind of use that to remember what a receptor protein does. They're going to receive a signal, right? Um, they're going to intercept a signal or intercept something from the outside, like in this example I'm about to show you. So here is some molecule. Maybe it's from a white blood cell and it's going to be a marker protein. Maybe in this case, it's just going to activate it so that it could let go of whatever substance holding on to, right? Maybe that's a hormone or a neurotransmitter. We're about to see an example with neurons. Those are the best examples of using receptor proteins. They do it all the time. I'll go over that in a moment. All right, but this had to receive some kind of signal, some kind of molecule in order to do its job and release whatever hormone or neurotransmitter this was. All right, here's a different kind of picture showing the same thing. The receptor protein here is going to intercept some kind of signal and release these messengers, which will carry out some kind of job inside the cell. Okay, and this is a GIF, I suppose, showing that process, right? So maybe the job of this receptor protein is to release this molecule called GTP so that it can go activate these other protein channels so that they can do their job, right? And so they all work together. So I said that I wanted to go over the example of a neuron when talking about receptor proteins because they do this all the time. They are constantly receiving signals. There's no point in your life when they are not doing this. Every emotion you feel, every emotion you feel, every time you have a thought, every time your brain has to convert a thought into words and then also make your muscles move in a certain way so that you can vocalize those thoughts, right? They have to send these signals from neuron to neuron, that's the type of cell, from cell to cell, right? And they do that. There's an electric pulse that goes down this long axon here, right? Here's the nucleus. Here's the Golgi apparatus, and the endoplasmic reticulum attached to the nucleus. Right, all that stuff is doing its job. An electric signal runs down the axon of this special cell, so that it can tell the end of it to release these neurotransmitters. Then these neurotransmitters are going to be released into the space between this neuron and this neuron. This space is called the synapse, right? And they're just going to move in random directions. And what did we talk about these past few days? About the properties of diffusion, how things diffuse. They will diffuse from an area of high concentration to low concentration, right? And so these will diffuse across the synapse and be received, intercepted by these receptor proteins on the other neuron. And then that will trigger it to send the next electrical signal to the next neuron, right? Until eventually your brain sends the signal down to your mouth, or to talk, or to your heart to beat, or your lungs to breathe, or whatever, right? The important thing out of all of that was you need to know what a receptor protein is. All right, I'll just going into unnecessary detail. All right. So here's another video that's going to explain what I just explained a little bit extra. So go ahead and watch that. I'll post the link in the Edpuzzle so you can see that. Go watch it. Come back here, answer the question that I have for you on that video. All right, and then move on when you're ready. The last type of protein that we're going to talk about today are called marker proteins. All right, so far we have transport proteins. They are going to transport materials into and out of the cell. We have our receptor proteins. They are going to intercept, right, receive signals so that they can release hormones, release neurotransmitters, and now finally marker proteins. All right, we should come up with another acronym like before so we can remember those things, right? So we have transport T, receptor R, and marker M. TRM. When we meet in class, we'll come up with some kind of acronym to remember TRM, TRM, TRM. All right, we'll do that together. Anyway, marker proteins. So here is a really terrible example of a cell, right? So here's the membrane. On the inside is mostly cytoplasm, which is made mostly of water, and the outside, right? So, and as with every cell, it's going to have some proteins on it, like this big red blob that you just saw appear. This is going to be our marker protein. Here's what their job is. They're going to have these little things on the outside, right? Boogeyman! that was a reference. So this marker protein, let's say it gets touched by a white blood cell. In case you don't know already, the job of your white blood cells is to kill bacteria, kill any foreign cells, right? So let's pretend for a second that you are playing a game, maybe, right? Let's say you're playing Manhunt, all right? I don't know if any of you ever played that, or Capture the Flag or something, and it's dark out. Usually you play this game in the dark, right? And you make your team, but because it's dark out, you don't really know who's on your team. Maybe you're playing paintball, and everyone's camouflaged, and you need to figure out a way to communicate with other people and come up with some kind of secret communication so that you know that they're a good guy, right? So maybe it's a hand signal, right? Or some kind of verbal thing. You have a secret password, Lollapalooza, whatever it is, right? And You now know that they're a good guy, right? Well, that's kind of what these marker proteins are. These marker proteins are a secret code so that the white blood cell knows that they're a good guy. All right. Otherwise, if the white blood cell comes up to something, a bacteria, a virus, whatever, and it doesn't provide that special that secret code, the marker protein, the white blood cell is going to be like, all right, you mine, right? It's going to gobble you up. Right. So they allow your body to recognize your own cells as being friendly. That's pretty much what I explained, but much quicker. And they're going to keep your immune system from attacking your cells. Here's an example. This should start moving in a second. There we go. So here is a white blood cell. And these white blood cells are going to use these antibodies. See this purple Y-shaped thing? That purple Y-shaped thing is an antibody. Usually you might take those as pills, right? Or hopefully your body develops them on your own to strengthen your immune system. That antibody is going to then have the special ends to it to attach to the bacteria and drag it into the white blood cell. It's going to go over it one more time, and I'll explain it as it does. Right. So you take a pill. That pill is an antibody. Your white blood cell uses the antibody to attach to the bacteria so that it can swallow it up. This is a process called endocytosis. We're going to go over that later. And it's now that it has eaten the bacteria, the lysosome. Remember that word? The lysosome in an organelle inside of the cell is going to do its job as the cleanup crew. It's all coming together now, right? And kill the bacteria, right? Anyone remember from first marking period talking about lysosomes and all the other organelles in the cell, right? Cool. And I think that's it. All right. So Ed Puzzle's going to end. Um, and yeah.